Hello, I'm Dr. Acker, and this is the lecture for the structure and function of the immune system for Module 2. This lecture is meant to provide you with supplemental information for your assigned readings and activities within the module. I would like to offer that this lecture is not all-inclusive of the concepts you need to master for this module, nor is it verbatim the information found within your text or supplemental learning activities that have been assigned. The immune system is the basis for all pathophysiologic concepts that we're going to discuss this semester. As the foundation for clinical assessment, decision-making, and management of diseases and patients that you may see in primary care. Now, the immune system is a very complex system, and its main job is to preserve life in homeostasis. The function of the immune system is to protect us from foreign organisms by distinguishing self from non-self organisms. Now, within the immune system, there are multiple structures and components that we're going to discuss. During this lecture, we're going to talk about the structures of the immune system that include skin and mucous membranes, mononuclear phagocyte system, the lymph node system, and bone marrow. Now, there are two components of defense within the immune system: specific defenses and innate defenses. Now, specific defenses are antibody and cell-mediated responses that provide a specific response in memory of a previously encountered antigen. This would be an example of a patient that comes to clinic with complaints of a peanut allergy. So let's assume it's a child, and he eats a peanut for the first time. It's introduced into his body, and the body is a little slow to pick up on it, and so nothing really happens. They have to form an antibody, basically a map of everything in the body, and they're mapping this new organism that comes in. Now, the cell-mediated response comes in after the antibody's been formed. The child eats another peanut, and that's when they have an allergic reaction, because the cell-mediated response is released by the antibody that was created the first time the child ate it. And therefore, your body now knows that this is a foreign substance, and it needs to alert all the other immune cells to come into play. Now, when we look at innate defenses, this includes things like your epithelial barrier or your skin, mucociliary clearance or your mucous membrane, phagocytotic cells, and complement proteins. Now, while the innate defenses don't have the ability to provide a specific response to an antigen, they're very important, though, because they provide natural immunity from many microorganisms. Now, natural immunity is an immunity that you might get from birth. The newborn baby has a passive immunity from the mother, from an antibody that she had in her body. So basically, in a nutshell, the immune system works like this. When a tissue is injured or a microorganism is introduced into the body, inflammatory chemicals are released and attract immune cells to the area to try to provide homeostasis again for that person. Now, when we look at the immune system, there is primary and secondary organs within the immune system. The primary lymphoid organs are the thymus gland and bone marrow. And the secondary thyroid organs include lymph nodes, spleen, and gut-associated tissue. So when we're looking at the primary lymphoid organ of bone marrow, this is absolutely the most important part of the immune system, as all the cells in the immune system are initially derived from bone marrow. They are formed through a process called hematopoiesis, and during hematopoiesis, bone marrow-derived stem cells differentiate into other cells, such as maturing cells of the immune system, or into precursor cells that migrate out of the bone marrow to continue their maturation elsewhere in the body. Now, Bone marrow produces B cells, natural killer cells, granulocytes, and immature thymocytes in addition to red blood cells and platelets. Now, we're going to talk about red blood cells and platelets later on in this course. As we look at the other primary lymphoid organ of the thymus gland is located behind your sternum and between your lungs and is only active until puberty. Now, many people confuse this with the thyroid gland, and they are not the same thing. We will talk about the thyroid gland later on in the semester. Now, the thymus starts to slowly shrink and is replaced by fat during puberty. Well, actually, right after puberty. And the thymus secretes a hormone called thymosin. And thymosin is really important in the immune fight as it stimulates and develops the disease-fighting T lymphocytes, or T cells that we'll call it for the rest of this lecture. Now, the thymus gland will not function throughout a full lifetime of a human being, but it does have a very big responsibility when it's active. It helps the body protect itself against autoimmunity, which occurs in the immune system when your body basically turns against itself. So an example of this would be a heart transplant patient that rejects the organ, or a Rh incompatibility when you're pregnant. So before birth and throughout childhood, the thymus is really instrumental in the production and maturation of T cells by secreting the thymosin that is needed for development and production. So kind of a cool deal about the thymus is that unlike most organisms, it's at its largest size during childhood. But, it shrinks over time and is replaced by fat. So when you're about 75 years old, the thymus is just a little fatty tissue. The cool thing is, the thymus produces all of your T cells that you need by the time you reach puberty. Now, the immune system is so vast, and we often think of it as a large system. In reality, it is a very intricate, detailed accumulation of many parts. And so I think it's really important that we start with the basics and build our way up. So when we look at cellular components of whole blood, that's really the basis, as that circulates all the blood cells within our body. So blood is comprised of erythrocytes, leukocytes, and platelets. We'll discuss erythrocytes and platelets later on in the semester, and we're really just going to focus on the leukocytes as they are the foundation of the immune system. There are 5 types of leukocytes within whole blood. They are neutrophils that make up 40 to 75% of your whole blood content. Eosinophils are 1 to 6%, basophils are less than 1%, and monocytes are 2 to 10%, and lymphocytes are about 20 to 45% of your whole blood volume. As a whole, these cells are also known as granulocytes because of the granules in their cytoplasms, and monocytes and lymphocytes are also known as mononuclear cells, and this was covered in Module 1 reading. So, when we're looking at the bone marrow, it's really important to understand how cells are differentiated. And the very first cell that we're going to discuss is the stem cell, and we're going to look at the differentiation of that. Now, I recorded this PowerPoint with Camtasia, therefore you don't have the ability to click the video buttons and watch this at your convenience. So I'm going to go ahead and click it for you. Now, if you're interested, I can send you the link to this whole video. It's about 18 minutes long, but for this lecture, we're going to watch only the first 5 minutes. So I'm going to go ahead and hit play, and we will watch this.
What's up, biology students? Your benevolent biology teacher here. In today's video, we're going to talk about how the cells in multicellular organisms specialize, and begin to perform specific roles and functions inside that organism. We call this process cell differentiation, and it all begins with these cells called stem cells. In mammals like us, this is where life begins. On this slide, we're seeing some of the earliest stages of human life playing out inside the female reproductive tract. This structure here is called an ovary, and this is where female gametes are made via meiosis. These gametes, called eggs, or more specifically oocytes, are released from the ovary into the fallopian tubes during ovulation. If the timing is right, sperm cells, the gametes produced by males, will fertilize this egg here in the fallopian tube. This fertilized egg, called a zygote, now contains a full set of chromosomes, 23 homologous pairs if we're talking about humans. As a result, this zygote contains all the genes necessary to build a complete human being, a full set of genetic instructions. As it travels through the fallopian tube, this zygote begins to go through the cell cycle, growing, replicating its DNA, and eventually dividing through a combination of mitosis and cytokinesis. From one cell to two, from 2 cells to 4, then 8, and so on. Eventually, this ball of cells that we call an embryo reaches the uterus, and if conditions are right, it will implant itself into the uterine wall, integrating itself with the mother so that the mother can nurture this developing organism using her own body systems. That's because this ball of cells has no body systems of its own. It doesn't have a nervous system, a digestive system, a muscular system, or any other systems. It doesn't even have nerve cells, or muscle cells, or specialized cells of any kind. All the cells that make up this embryo are completely generic and do not have any special functions or features. However, all of the specialized cells that make up our bodies, our muscle cells, nerve cells, skin cells, bone cells, blood cells, and so forth, all descended from an embryo just like this. So how do our cells come to take on these specialized roles? How do muscle cells become muscle cells? And what keeps them from becoming nerve cells? That's what today's video is all about. We call undifferentiated, unspecialized cells stem cells. These cells have the potential to become many different kinds of cells. As these cells divide and replicate themselves, they go through a process called differentiation. And it is through this process that cells are able to specialize and take on specific roles and functions in the body. Totipotent stem cells are the most flexible. They have the potential to become absolutely any kind of cell in the body because they are completely undifferentiated. Zygotes are totipotent cells, as are all the cells in an embryo up until that embryo reaches the 16-cell stage. After this stage, the cells in that embryo begin to differentiate and go down different roads of development. The outermost cells are destined to become the placenta, a structure that nurtures the developing embryo by integrating itself with the mother's tissues. And that's all these cells are ever capable of being from here on out. The rest of the cells in that embryo will grow into a new human being, and we call these cells pluripotent stem cells. Pluripotent stem cells are still mostly undifferentiated. They can become any cell in the body except the cells that make up the placenta. As these pluripotent stem cells continue to grow, develop, and divide, they continue to differentiate and become more and more specialized and less flexible as the differentiation process pushes these cells down different developmental roads. Even as adults, our bodies still contain undifferentiated stem cells. However, as adults, these cells are devoted to a particular cell lineage and are somewhat limited in their ability to differentiate into different kinds of cells. We call these adult stem cells multipotent because they are still able to become several different kinds of cells, but they are no longer unlimited in this ability in the way that pluripotent embryonic stem cells are. For example, a multipotent stem cell might be devoted to the blood cell lineage. This cell has the potential to become any kind of blood cell, white blood cells, red blood cells, platelet cells, or cells of the immune system like B cells and T cells. However, these cells will never become muscle cells or nerve cells because they have already differentiated beyond their ability to become those kinds of cells. So this is cell differentiation, by which undifferentiated stem cells become more and more specialized. But what controls this process? What makes some cells go down one road while other cells go down another?
So that should be a review from your undergrad patho course. If it is not, be sure you really, really study that because you may see that later on in the course in quizzes and final exams. So, another really important feature when we look at how everything comes together is to discuss immunoglobulins. So, immunoglobulins also are known as antibodies. They're glycoprotein molecules that are produced by plasma cells, which are white blood cells, and they act a very important part in the immune system by specifically recognizing and binding to particular antigens such as bacteria, viruses, and microorganisms that are introduced within our bodies, and they aid in their destruction. Now, there are 5 major antibody classes. This is a very important concept that you're going to need to know inside and out for the remainder of our course and again in practice. There is IgG, IgM, IgA, IgD, and IgE. We'll discuss these in great detail all the way throughout the course as they apply to labs that we may order or disease processes that we are learning about. Now, with immunoglobulins, as they are antibodies, we must talk about passive immunity and active immunity. So passive immunity is an immediate but temporary protection from a microorganism. So for example, mothers that are immunized with the flu vaccination, let's say in November, if they deliver a few weeks down the way, that immunization has the ability to cross the placenta and it will cover the baby for a few months. Now, we know that this is not a permanent immunity or an active immunity because the baby has to get revaccinated the following year. Now, active immunity is when people are exposed to an antigen that stimulates their own lymphocytes to produce memory cells. This is a long-term immunity, but it takes weeks to develop, such as if a patient had acquired hepatitis A, their own lymphocytes would have made memory cells, sort of like a roadmap, identifying that hepatitis A, and they're able to build their own immunity. So that is really the difference between passive and active immunity. So as we're moving forward into the immune system, we're going to look at the major cellular components of the immune system, the leukocytes or white blood cells. So within this, we have monocytes, macrophages, lymphocytes, and then the granulocyte cells, which are neutrophils, eosinophils, and basophils. So when we look at this, leukocytes are the core of the immune response of the immune system and response. So monocytes are larger than any other leukocytes within the system, and they mature into macrophages once they're released into the bloodstream. So a monocyte will migrate to tissues, particularly the liver, lymph nodes, and lungs, where they may actually stay for days or years. Once they're released into the system, macrophages develop, and they participate in antigen processing, tissue repair, and secretion of mediators for a specific immune response. They're commonly found near mucosal surfaces and take the organism to the secondary lymph node organs. And remember, we talked about those earlier, and those would include lymph nodes, spleen, and gut-associated lymphoid tissues. Excuse me. So when they're taken to those secondary organs, they're processed, and then it's determined the antigen for the discernment of the T lymphocytes. They also act as effector cells for some types of tumor immunities. So monocytes are generally sent to sites of inflammation where they mature into macrophages. So the epithelial, I always say this wrong, dendritic cells basically have the same function as a macrophage. However, their focus is on processing and transporting antigens from the skin, respiratory, and GI surfaces to the regional lymph systems as opposed from the mucosal um, tissues. So as we look at monocytes and macrophages, um, we talked about that monocytes are larger than other leukocytes and that they mature into the macrophages. And we talked about how macrophages, it deals with antigen processing, tissue repair, and secretion of mediator for specific immune responses. They do this by a process called phagocytosis, which again should be reviewed for you. Now, macrophages make up 3 to 8% of the total white cell count. The percentages we talked about earlier were of the total whole blood, so this is just the white count part of it, okay? So there is a little 2-minute video that actually shows a cell undergoing phagocytosis. And so again, I'm going to click this for you to watch as you're not able to click it. And we'll just watch this really quick. So that is phagocytosis, and it's pretty amazing that that's going on in our bodies all the time. So as we move forward and we continue to look at the components of the immune system and the leukocytes, we're going to focus on lymphocytes. So lymphocytes are responsible for the initial specific recognition of an antigen. Once lymphocytes mature, they move to the secondary lymphoid structures, the spleen, tonsils, lymph nodes, and that is where this next process occurs. So there's two main types of lymphocytes. They are B cells and T cells. Now, B cells are characterized by the presence of immunoglobulins on their surface, and upon the stimulation with antigen, they're then transformed into plasma cells. Now, plasma cells are able to secrete antibodies specific to the antigen. So B cells are developed in the bone marrow and are involved in humoral and antibody responses. They are the antibody producing cells. Now, T cells develop in the thymus and are involved in cellular immune response. So the T cell takes part in the cell-mediated immune response, which does not depend on the presence of circulating antibodies, remember. Now, there are numerous types of T cells circulating in our bodies. However, we're only going to focus during this lecture on just a few. And some of them will sound familiar, and we'll delve deeper into them as we look at disease processes throughout the semester. So the first T cell that we're going to talk about is the helper T cell. This is the CD4 cell, often known in HIV and AIDS reporting results that are reported. Now, the CD4 cell helps increase B cell production of immunoglobulin, and it amplifies the T cell mediated cytotoxicity. By doing this, it helps to regulate immune responses through cell-to-cell contact. Now, the CD4 and CD8 cells are both cytotoxic cells. Now, when we look at cytotoxic or killer T cells, which are CTLs, they are created after mature T cells interact with certain foreign antigens. They are responsible for defending our bodies against viruses, tumor immunities, and organ graft rejection, like we've talked about previously. Now, CTLs may kill their target through osmotic lysis or induced apoptosis. Now, B lymphocytes plays a major role in the differentiation to the antibody-secreting plasma cells. They may also release cytokines. They function as more of an antigen-presenting cell. So all mature B cells have surface antigens, or Ig. So that's the first part of that immunoglobulin. Okay, they all have those. And that acts as their antigen-specific receptor. Now, the most common receptor in our body is the IgG. Okay, and again, we're going to learn more about all of these immunoglobulins throughout the semester as we're talking about other diseases. But it's a really important concept to understand. The last T cell that we're going to talk about are natural killer cells. Now, they have a very distinct cell surface marker, but they lack that antigen-specific T cell receptor. Therefore, they have to work with T cells to be effective. They cannot work on their own. So natural killer cells migrate to the site of inflammation, and they cause cell-mediated cytotoxicity through cytolysis. Now, other functions of this cell include antibody-independent cellular killing, apoptosis, and immunodeletion of responses to viruses, malignancies, and transplanted tissues. So they serve a very, very big role that's very important in the immune system. As we look at neutrophils, they're kind of interesting. They also help the immune system through phagocytosis, and they also make up 60 to 80% of the total white blood cell count in the body. The cool thing is that these are pretty fast acting. So within 12 hours of being released from the bone marrow into the bloodstream, neutrophils migrate into extravascular tissue. So tissue neutrophils are activated by what's called a chemoattractant. And a chemoattractant is like what's released from the antigen in the system. And at the site of injury is when they're most abundant. And so basically your tissue gets injured and it releases this chemical that says help, 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 and the neutrophils come running and they see what they can do, and if they need to elicit a response, they can do so. However, they really work by, they're your first line, so they work by ingesting that bacteria by phagocytosis, and then they also release this enzyme, which is called lysosome, and that actually destroys the bacteria. So the next cell we're going to talk about is the eosinophils. They are cytotoxic, not phagocytotic, and they make up 1 to 6% of your total white cell count. So eosinophils migrate from the marrow, bone marrow, through the blood into extravascular spaces and tissues, and they survive there for weeks. They are found in inflammatory sites and works against parasites. That is their main role. If you have a parasite, the eosinophils are the ones that are coming out to fight that. When activated, eosinophils release several inflammatory responses, but one in particular, the MBP, which is major basic protein, is what destroys the parasite. It does this by impairing ciliary beating and causes exfoliation of respiratory epithelial cells and can also trigger a histamine release from mast and basophils. Now, when you think about that, if you have a antigen that comes in that gives off an allergic response somewhere, then the MBP, not only does it attack it and keeps it from growing, the side effect is that it exfoliates respiratory epithelial cells. So, can you think of a condition that this may play a factor into exacerbating? It may help kill that parasite, but it also makes other conditions worse. And that condition would be any sort of airway hyperreactivity, because as it's exfoliating, it's loosening up those respiratory endothelial cells. They're just vast and loose. That asthmatic patient or that reactive airway patient is going to have an attack. So again, the movement of eosinophils, like neutrophils, are phagocytotic in the sense that they are able to release responses that will kill a parasite, but they do not actually engulf. They do not ingest the organisms, but they do exert that cytotoxic effect on them. All right, moving on to our last cell that we're going to talk about in this lecture is basophils. So they are phagocytotic, and they make up 0 to 2% of the total white cell count in your body. So basophils' important role is in both the immediate and latent phases of allergic responses. They release several mediators of allergic inflammatory diseases such as histamines, leukotrienes, and prostaglandins. Now, in many allergic reactions, the immune system, when first exposed to that allergen, produces a type of antibody called, can you guess? IgE. So you know that all of those antibodies have to be an Ig. So we just have to figure out which type. IgE binds to the type of white blood cell, primarily basophils, and they also can attach to mast cells in the tissue. So the first exposure may make the person sensitive, like we talked about before, to that allergen, that child with a peanut allergy, and causes no symptoms. But when sensitized, that child will subsequently encounter that allergen, and the basophils and mast cells with that IgE on their surface release the substances, histamine, prostaglandins, leukotrienes, that can cause swelling and inflammation in the surrounding tissues. So as we bring it all together, you know, the immune system is just fascinating to me, and it is the basis for maintaining life, and having a thorough understanding of the immune system is really important to pathologic concepts, as when we're learning about disease processes and treating patients, it's really all going to start back at the cellular level. So if you can grasp this information that you're getting in the first couple modules of this course, you're going to be so far ahead when you move forward and you start seeing patients as you understand their disease processes. You'll find throughout the remainder of this course that every process we cover will involve the immune system at some level. So while this lecture is certainly not all-inclusive, be sure to review the remainder of the assigned readings and activities for this module, and also be sure to review your course objectives for the entire course to make sure you're staying on track. So I hope this lecture was helpful, and I look forward to a great semester with you.